Okay, so let's get started. Once again, hello, welcome, and thank you for joining us for today's webinar. Dr. Salvador Gezan will be presenting, and he will discuss how to partition additive dominance and epistatic genetic effects using an animal model with ASReml-R. All right, so without further ado, I'll hand you over to Salvador now.
Hello, I imagine you can hear me there. So thank you for joining again for this webinar, and I'll talk about a slightly different topic. This is about partitioning the additive dominance and the epistatic. It's something that you probably have heard here and there in some papers. This is an old topic. Really, it's an extension of the animal model. But I think it becomes a bit more interesting with all the genomics data because it brings a little bit of more possibilities to explore it. So the way I'm going to go is I'm going to give you a little bit of theory for a generic example based on pedigree. And then I'm going to develop the example in R using ASReml-R. And then I'll come back to show you a few of the extensions with the genomics data. So, and in between here and there, feel free to ask questions and put some things in the Q&A, and then obviously we'll try to answer as much as possible. And this is not totally a simple topic, but it is quite interesting and relevant. So you're probably familiar about trying to partition the phenotypic response into a phenotypic value, a genetic effect, and an error. Again, this is all additive. Which has its own limitations, but we have been done tremendous progress with this model that is based on addition of effects. Obviously, once we partition the genetic effect, we can further partition that into the additive, the dominance, and the epistasis. And all those three elements, the A for additive, D for dominance, and I'm using an I for epistasis. Once they are estimated as effects or estimated as variance components, they allow us to understand more in a better way and in a better strategic way for our breeding programs on how that total genetic effect is partitioned. This is not something that is very easy. Requires a specific data, and particularly in plant breeding, this is a little bit easier because what we need is we need to test the progeny of a specific crosses of parents, and in this case, parents have to be crossed with, a male has to be crossed with several females and a female with several males, unless you have a nice deep pedigree that has crossed different individuals through the past. But also you need clones, and these clones need to be replicated. The reason for this replication is because we need to split the genetic effect from the error effect or from the residual. So they are quite demanding on the types of data. One thing I need to mention, and probably those that work more in plants are more familiar with it, when we estimate the additive effects, we are referring as estimating the GCA. In this case, is the breeding value or the additive effect is going to be twice, the breeding value is twice the GCA. When we talk about estimating the dominance, we talk about the interaction between loci, but in this case, often is as the interaction between two parents, their specific interaction that they produce on the performance of the offspring, also commonly known as SCA. And whatever is the rest, which is this high order interactions, and this is what happens when you have a specific combinations of alleles across different loci. It is that high order interaction that is very difficult to model, is what we observe in clones. And again, you're probably familiar that some clones are much better than the individual parents or the individual family. So we'll talk about this partitioning, but we actually focus on the variances associated with that partition. So we have a total genetic variance, which is what we use to calculate the broad sense heritability. And once we do the partition with a linear mixed model, we will have a portion that refers to the additive, which is our narrow sense heritability, a portion associated with the dominance variance, which is called D squares or dominance ratio, and a portion associated with the epistatic, which in this case is going to be the I square. And you can see here some of those variance components will lead to our generic variance. And obviously we have a specific gray part here, which is our residual part, which we can model, we can't extract, and that again is part of the noise. Now, what we do is when we fit this model is we'll put a specific components, animal component, maybe a family component, a clonal component, and so on. And those components are associated with the actual genetic variances that we are and of our interest. So I'll come back to that. So just to this is our genetic animal model. This is an animal model that only has an additive effect. And there might be a few fixed effects. Imagine a rep, some random effects, imagine a block random, or a plot effect, or something else that you may have. And the only term that you have here is the additive term, which is associated with an A matrix, which is the pedigree-based relationship matrix. And then obviously you have the error. Now notice that the error has the other non-genetic components. It's going to have the dominance, the epistasis, and it's going to have a true error, which is the error besides the genetic errors that you're not explaining. So that's why I'm calling this sigma square star, because in reality that sigma square has a little bit more genetic elements that we are not considering. We can calculate the heritability, no problem, by taking the variance associated with the animal divided by the phenotypic variance, which one definition here is the sum of all the terms. Now, notice that we can replace this A matrix by a genomic matrix, Van Reyden or Yang, or another form that we calculate based on molecular data, but I'll come back to that later. Now, the typical clonal model, which I know many of my friends from plant breeding are familiar with it, when you deal with clones that are replicated or sometimes recombinant in bred lines or double haploids and so on, we're dealing with individuals that are replicated as clones, so we can split the genetic effect from the error. And the model is basically the same as the additive model, but when we do with the G, we obviously don't have a relationship matrix associated to it. We just have an independent matrix, and the variance associated with G is going to give me the variance associated with the broad sense heritability. Notice now that this error has no genetic components at all, because all of them are absorbed into the G. Now, this is the key of it. What's the additive plus dominant plus epistatic model? And there's many names for it: additive plus not additive, full clonal model, and you will see it with different names. I'm going to use one of the approach based on an extensions from the animal model. There is another approach based on the extensions from the parental model. And the genomic models are a little bit different too. So again, same things as before: rep, incomplete blocks, or whatever, additive effects, which are associated with sigma square A. And there's this family effect that sometimes is male by female, but you have a column in your data set which is denoting the family, and that's the cross. And that family effect is going to be my SCA, and you assume they're independent and they're associated with a variance, as is called in this case sigma square fam. And then I have this last term, which is clone nested within family. You can call it clone nested within family, or you can call it the same thing as the individual if you're using the same names. But notice that this one is not associated with any pedigree or associated with anything. It's just independent. And these are the epistatic errors, plus a portion of the elements that is not being explained by the family term, okay? And then you have the pure error that it comes through it. So when we do estimated the additive variance, and this is very important, we just say BA equals to sigma square A, BD equals to times sigma square family. And this is important. So the sigma square family is only 1/4 of the dominance. And that is because it's a specific interaction of males and females. So it's only 1/4. So you need to multiply by 4 to get the full dominance variance. And that also implies that the CF variance has 3/4 of the family variance that it wasn't extracted by this family term. And that's when you try to calculate the epistatic, you take the sigma square of the clone minus the sigma square A minus 3 times the family. And that is going to give you your actual term, to your actual epistatic component, okay? So we'll come back a little bit more to it and then explain a bit more details, but then that allows me to estimate heritability, broad sense heritability, narrow sense heritability, dominance, and epistatic, okay? So let's go to the example after all this, and then I'll come back to explain a few of the things. Now, the data comes from this paper, which I just pull up. It's publicly available. I don't understand all the details intricated with how the data comes, but it presents a very good opportunity for us to explore what happens. So I'm going to the model, and I'm going to just walk you through it. I need to read ASReml. And I'm going to read a few variables, ASR Genomics, HGH matrix to build some of the matrices and show you some things, and a couple of additional libraries to do some data manipulation. So this data comes from Strawberry in California. This is the reference. And I have, it corresponds to 574 genotypes. And this is over 2 years. But I'm only going to use the data for one of the years, which is year 1. And I had to do a little bit of renaming of columns and so on, but just to make things a little bit more easier and consistent. When I look at the individual traits, I found some outliers. So this is what I'm doing here. Some of the outliers for some of the traits, which are actually, let me show you, head of clonal. So you can see the columns associated with this. We have the ID, a block, so this is replicated 3 times, and different response variables, yield, count, weight. I'm only using 1 year, and I have the details from the mother and the father and the family associated with it, which I might use or might not use. Particularly, I'm going to use the family and obviously the entry ID, which is the specific clone. Recall that each of these clones is replicated 3 times because there's 3 blocks, okay? I found a few outliers on the response variable weight in case we want to explore it, and also one in count, but we don't need to worry. The main thing now is to make all those elements factors, and I'm going to move a little bit here. And so I'm making the mother, father, family, the entry, and the block all factors, elements that I think I might need to be used as factor later. And the first thing that's very important on these experiments is that in order to estimate, to separate the genetic effect, in this case, the total clonal effect from father, mother, and family, and this is also goes for full sieve analysis, is that you need to have good crossing. And that means that the father has to be crossed with a couple of mothers, the mother has to be crossed with a couple of fathers. And obviously you can't have just one clone per family, because that means that clone will be totally confounded with the dominance of that family. So you need several clones per family. Okay, it doesn't have to be a perfect data set, but here I just requested a query of by family, which one is the mother and which one is the father, and the number of observations. Recall, there is 3 blocks, so everything is multiplied by 3, but you can see here, for example, family 101 is the cross of these 2 parents, and it has 24 divided by 3. That gives me, it has 8 clones in there. Many of them tend to have 1. There's a few that have more than 2, 3, 4, 5 clones. So there's a good number of clones. The other thing, I'm going to sort by father. And you can see this father 02 is used in 2 crosses. This father 05 is used like in many other crosses. And the same thing you can check with the mother. The mother 04 is used, for example, here, 04 is used in 2 crosses. So this seems to be a good connectivity in order to separate some of the things. It's not perfect, and many of these clones are actually going to have elements confounded, okay? If you have questions, probably it's better that you post your questions on the Q&A, and then I'll be able, once I find a good spot to stop, I'll go over the question, and also I will answer the questions later on by a long Word document that it will make available for everybody, okay? So let me just go over a few things. So other checks, check of the phenotypic response. In this case, I'm going to use yield divided by 100 just to make things a bit easier. It doesn't look too bad, just looking for outliers of very extreme things. And then you have, this is a plot with all the different traits in case you want to explore, and clearly there is a few little outliers, okay? So let me just go to the basic model. The basic model only has the genetic effect and the block. There's nothing else. There's no other random effects. And this is going to allow to have the total genetic value. So if you fit it, fixed effect yield plus block, random entry ID, totally independent. So this is going to give me the variance of G. And then the errors is just a simple error term. So we do look at the plot just to check if potentially there is some outliers. Potentially there is something here and there, but I'm not going to worry. And this is the variances associated, 6.4 for the total genetic variance, 1.43 for the error, and this is fixed. It's just something from ASReml. I calculate the broad sense heritability as the ratio of the first one against the sum of the first and the second one, and we have a tremendous heritability on this particular data set, 0.87. And imagine that heritability is because we have very extreme clones from very good to very bad, and that implies that we have a tremendous heritability. Depending on the study where you're dealing with, if you're in forestry, sometimes you have values of 0.6, 0.5, and you're probably familiar with your crops and having other values. But for me, this is a little bit on the large side. And then I can get the total genetic values by using the summary. These are going to be the BLAP effects. They are deviations with respect to the mean. And you can see here a few of them that happen to be negative. So 3 units below the mean, 1.4 units above the mean, and so on with the corresponding standard errors, which is associated with the number of replicates that they have. And we can get predictions, and predictions is the same thing, but it's just we're adding the overall mean. And this works very well for these models, the predictions, but once you deal into all this partitioning, you'll realize that it's not as easy. Okay, so that's good. So I'm going to read the pedigree. Now, this is very interesting because this actually manuscript has the actual pedigree. And the pedigree, I'm going to read it. It's a file. It's usually constructed with these 3 rows. So I have the offspring, the parent A, and the parent B. There's no distinction between which one it is. And the data, which also is a binary trait that says this is present or not in the data. This is just to help a little bit. But there's one thing that is very important on this pedigree, which has actually 15,656 observations. So there's a lot of generations and a lot of records. So this is a whole full historical pedigree. We can calculate the A matrix, which I'm going to need, and this takes a little bit more time. And one thing I'm going to do is I'm going to show you the kinship heat map from this data set, just to check a little bit of the connectivity. Notice that the A matrix is being calculated with the AGH matrix library, and I'm filtering here only for those individuals that have data, okay, just to see how they are, because the whole matrix is extremely large to show. And then I'm using this kinship heat map just to show you what happens with the data. It takes a little bit of time, but only a little bit. While we wait for that, there's this function from ASReml to give me the A inverse, which I'm going to use directly from the model. And I'm going to need that, and we'll just see a few of those lines, okay? So that should show really in a couple of seconds. Sometimes it takes a little bit longer. It's computing, and then should be showing me the kinship heat map in a sec. So here you see the heat map. I like to observe it. You don't want this to be all red, sorry, all blue, which means there's no relationships, and just a few spots where you have the values that are closer to 1 or something. You want something like this, which means all these different individuals that are being tested on this specific phenotypic data set have connections through maybe lots of generations behind. Maybe that's some family structure. We know some of them share parents in common. You can see it here. So it looks very good for allowing us to separate additive effects from non-additive effects and so on. Obviously, it will depend a little bit more on the structure of the data that we have. And this is what we're going to explore now. Okay. So I'm going to, again, just look at my data. I'm going to create a copy of the entry ID, and I'm going to call it entry additive. This is just a little bit to assist ASReml to separate between the effect that it's the proper additive effect from what it is the genetic effect from this, the epistatic effect from this clone, or the rest of the genetic effect. So in this case, I'm going to fit this model, and notice that in difference with the additive, with an additive model, I have BM, the entry ID I associated with the A matrix, and then in this case, the A inverse. Then I have my family ID term, and I have this IDV entry ID. And I'm just being very specific on saying that it's an identity with a single variance. You can drop that one if you don't want to, and that's okay. You have exactly the same model. But everything seems okay. Now it has converged, 1822 is going to be a very different value of likelihood than the previous model, because now we are separating more terms. And actually, you see a tremendous improvement for the same data with respect to the other model. The plot will help me to assist. It has a very similar structure. Now, this is where things become interesting because we have the different variances associated with different terms. Now, this is the family ID. We multiply by 4 and we'll get the dominance variance. And then if we divide it by the total phenotypic variance, we'll get the dominance ratio. This term here is the additive variance 3.5, which is good. And then we have this term, which is the rest of the genetic or the non-additive. It's 3/4 of the dominance plus 3/4 of the dominance plus the epistatic effect. So now I'm in a position to calculate different components, and these are the broad sense heritability 0.80, which is very similar to the previous one that we obtained when we fit the model without partitioning the components, and it's good to check that it gives you around the same. So it's 0.87. Topper is 0.817, and then in the new analysis, I'm obtaining 0.80, and a relatively small standard error, which is great. Then I have the additive variance, or in this case, the narrow sense heritability. 0.48 is a good number for these studies. I'm familiar with the strawberry, so this is reasonable for a strawberry for yield. There's relatively small standard error, not too small, but relatively small. Then we have 0.21 for dominance, and then we have 0.10 for epistasis. So it's all this very good. Now, why this is very important? The standard errors are all around 0.10 in this, which means this might be 0, this might be changed a little bit. You not usually have enough information for all of this. It requires a good data set. But the fact that these are not, let's say, 0.3, it leads me that I'm able to split these terms successfully. There are other measures, but it tells me that I'm able to split these terms successfully with this data. Now, sometimes the system crashes when you analyze this, and this might be for other reasons, such as, for example, you have single parent crosses, a male cross only with one female, and your pedigree is not very deep, so the estimation of, for example, the family ID is not very good. Or where you have too few copies of the or clones, then there is hard to split the clone from the error term, and so on. So you will get them that with something that doesn't converge or with very strange results. Sometimes you get negative epistasis, and a negative epistasis is very common because you get some of the other variance components like dominance or additive effect inflated. And when you get them too inflated, it means there's nothing left for the epistasis, and it tends to be negative. And I can show you a little bit what happens, for example, if I use, I think it's count. If I use count, it's the count of number of fruits. You will calculate all this, and you see this epistasis to be minus 0.13 with a very large error, which says it could also be 0. Everything else is a little bit higher than before. Okay, but I'm going to go back to my original model with yield. Yield divided by 100, and then the variance components. And there's a couple of things that I want to mention that are very important that I probably will mention more later, but it's the interpretation of these components. So for example, one of the things is that if you have the majority of your genetic variance in the epistasis, it's a kind of like a problem, because that means you need to select the best clones. And the only way to identify the best clones is by comparing clones individually. Now, if the majority of your genetic variance is in the additive part, that means if you select the best parents, you're probably going to find the best clones eventually down the line. Because by selecting the best parents, which is similar in this case, you're capturing the majority of your genetic variance that you can capture in your selection. Now, let's say if your majority is in the dominance, that means, or there is a good portion of the dominance, selecting the best family will be very relevant. Like in this particular case, 0.48 plus 0.21 gives me 0.70, more or less, which means I capture almost all my genetic variance by selecting the best families. So if you just focus on selecting the best families, and maybe a minor interest on selecting the best clones, you will end up with the best possible genetics on this particular trait, okay? So it leads really to decide what's the best for a breeding program. So as friends that are in forestry, and I work in pine before, normally we have a really good additive variance and relatively small dominance and epistasis. For example, we have 0.48 and 0.10 and 0.10. So then we say, well, our focus means really should be in selecting the best parents for the next generations rather than try to get the best or identify the best family and identify the best clones within family. And in some crops like pines, it's difficult to get propagated individuals, even though sometimes you can get them. So you might rule that out and you say, I'm only losing 10% of the total genetic on the total phenotypic variance on my selection, but I'm saving a lot of costs. So there is a lot of different things in there. Okay, there's a couple of questions. One of them is the fitted versus residual plot shape in a strawberry DSMI indicating heterogeneous variance. Do I think this is related to the broad sense heritability? No, I don't think it's related. It could affect a little bit of the heritability. And yes, it's a little bit of heterogeneous variance here. Maybe a transformation will help. But you have to be a bit careful with this because there could be many other factors that are influencing this. I mean, for example, special components are being missing here, and that also could influence. Now, somebody's asking, why did you not use the college heritability? And my answer is, well, the college heritability is good to assess the reliability of each of those measurements. But in this case, I'm focusing on the partition. So I'm focusing, and it's not going to give me the same measure. And also notice the college heritability works on the mean value of the phenotypic response. In this particular case, because I'm using the error variance, I'm looking at the actual partition based on the individual clone that I have here. I could divide this V4 by 3, and I will have the mean broad sense heritability, the mean narrow sense heritability, the mean dominance, and the mean epistasis. But in this case, this is giving me a bit of the partition. One thing that is very important is that these variance components can allow us to also design experiments in a different way, okay? So they allow us to, they might tell us where the main thing is, and maybe what we need to do is, let's say, focus on families with fewer replicates because we don't have too much error variance, or we have to focus on more clones with fewer replicates, or in fewer clones with more replicas. You know, all of those combinations, this is the sort of information it gives. And by the way, this information is also very important to assess a breeding program and what is the best strategy. Do you focus on additive effects? Do you focus on family effect? Do you focus on clones? And it's something we use a lot for when we are focusing on improving the breeding strategy and the testing of a breeding program, okay? I'm going to move a little bit along. These are BLAP effects. The BLAP effects is going to be a combination of different effects, in this case, the family effect, the additive effects, and so on. So I'll come back to those a little bit ahead, okay? Now, I have a few comments. I already talked about the interpretation of this, and every one of these effects, it has something important, and this is what I'll show you a little bit ahead. And then you have to be careful with the use of the predictions. And this is very clear because I might want to take the family prediction, but notice that the family prediction has only the SCA, or if you have entry ID prediction, you're going to have been contaminated by the family ID. So be very careful when you use the LS means of the predictions associated with this. It's better that you cannot construct them yourself. Now, before I go back to look at those specific effects, what I'm going to do is I'm going to fit the model now, but only with the A part, only the additive portion of this model. And what am I doing this? Is because I want to show you something very interesting that happens with this model. This is my original model where I have the additive effect to be 3.5 and error part 1.43. Now, if I drop these two terms here, which I don't have it on this model, I expect them to be added to the residual part if everything is orthogonal. But what you see here is that they're not really totally added. into the residual part. And notice that the additive effect changes to 8.57. So the additive variance from being 0.48 on my previous analysis changes now to be 0.77. And this is a concern because that means that 0.77, it seems to be inflated by the other genetic effects. And that has tremendous implication because your breeding values are going to be inflated, your genetic gains are going to be inflated, and so on. So you're really, and this is something very common when you don't have a very good structure of crossing and things like that. Now, I don't know more details about this population that could explain that and why and what was tested here. But notice that there's a lot of families that had only one clone, and that's potentially the problem. That is not very easy to separate it once you use an additive model specifically for this. Now, if you replace the pedigree by a genomic matrix, you will be able to fix that a little bit better. But in this particular case, it doesn't seem very good. Now, a couple of more things. The model, the full model is actually statistically much better. The log likelihood is much smaller than the other model. And then the likelihood ratio test that we have here, it tells me that these two models are significantly different, okay? So again, I'll continue a little bit to show you what happens here, and then I'll stop for a few questions, okay? Now, I'm going to say there is another model which is quite a nice alternative, which is the good between the two worlds. Notice that in this model, I have additive plus non-additive effects. So I have the VM, which has the sigma square additive. And then I have this IDV entry ID. Because I don't have the family term does not get absorbed into the error, gets absorbed into the rest, which is these non-additive effects, okay? And then when you get this model, then I'm saying I'm not going to do the separation between family and epistasis. I'm just going to clump them together. And that often is a good strategy when you don't have the best possible crossing between things. And also, if your focus is not too much into the non-additive effects, but you still want a model that gives you additive effects that are improved, then you fit this model. It's usually going to converge fine. Whatever mysteries happens on this non-additive part, you don't care too much. They're just sum up. The model converges very well. I'll show you. This is the original additive model, 8.57, quite inflated. The full model that I showed you before is 3.50 for the additive component. And then once I fit the non-additive, additive plus non-additive is 3.49, very similar to that one. And actually 2.24 is the sum of these two. And then you can see here that it's very safe model and the error term is fine. And the correlation between the breeding values, which I'm not doing here, is going to be very similar, okay? So that's a little bit what happens. You can see the gross sense heritability of this new model that has the two non-additive is almost 0.80. Then the narrow sense heritability for this non-additive model, it's 0.48, which is very similar to before, and then 0.31. Now, sometimes I fit these models, comparing them one to the other, so you can see a little bit if you have some very strange behavior that what's happening. Because not everything is perfect. Sometimes you see that your model with non-additive looks very weird and leaves you with the additive variance to be close to 0. And that means it's being absorbed into all the other terms instead of being back and so on. So it is a good assessment. And this is, again, because these models are not perfect. Log likelihood values, this is the model with the additive, clearly worse than the full model. And look at the model that doesn't separate the dominance from the epistasis. It's not very far away from the full model. It is significantly different, but again, the difference is not very bad. I'm just using some criteria here. If you use the AIC to select the best model between those three options, you will find that the full model is the best option. If you use the BIC, you'll find that the best model is the one that has the non-additive effects, okay? I'm going to answer a couple of little questions. Sorry, the best model in this case is the one that has just the non-additive effect combined. So somebody's asking if in fish, as a male mate is to 2 or 3 females and vice versa, can the family serve as the same purpose? Yes, you need to know that the kid belongs to one male or to the other female. So that is structure of 2 by 3 or even sometimes 2 by 2, which means a male cross with 2 female and a female cross with 2 male, that even if it's a unit block, it will work fine. Especially if you have some pedigree over generations above. So it works perfectly. I use it in a salmon breeding program that I consulted for. Somebody's asking about LS means. This is a least square means. It's not the same as the blues. The blues is the effects for fixed effects. The LS means is something that is used from SAS. It means that you're estimating the means based on the fitting of your model, which is LS least squares. In this case, is REML, so I should probably call them REML means, okay? Somebody asking about multi-location. So for multi-location, you obviously have to just create the additional dimension of adding the sites or the location. So site effect, block nested within site. Then you have to have the genetic effect, the main genetic effect if you want it, and then the interaction of genetic effect with additive, the main family effect, and then the interaction with family ID, and also for the non-additive. In those cases, probably this model that has additive plus non-additive will work a little bit better and safer, because not only you need to make sure that the parents are crossed, like one male with female, with 2 females and a female with 2 males, you also need to make sure that the individuals, in this case, the clones on the families, are tested in multiple locations, not only in one of them. Otherwise, you cannot estimate very well what happens with it. So there's another dimension that I'm not expanding here, okay? So I'm going back to this model just to show you, and not much time left, so I want to be able to present everything. So this is my full model. I'm relatively happy with the full model. It's still a bit concerned when I don't put the additive effects, in there. Sorry, only the additive effects. But again, that needs a little bit more exploring and understanding of the problem. We look at the different partition of the elements, so I'm happy with it. It makes sense from what I know from the population and so on. So what I'm going to do now is I'm just going to walk a little bit to get the breeding values to make my decisions for breeding, okay? My decisions from this data from this particular model. I just put the mean of the population here, 5.34, just to use a little bit of a reference. So you'll have to trust me with some of the manipulations I do. So I have, in this particular case, a data frame just with the additive effects. This is the this is the pedigree-based breeding values. You can see them here. And actually, I can select and I calculate the reliability, which I find it very relevant. You have some individuals that have really negative reliability, which is a bit strange, but that happens, is because they have such a high standard error because there's no data from them. By the way, these breeding values is a set of 15,600 plus breeding values, okay? So it's a lot of them because everybody who is in the pedigree gets a breeding value. Now, if I rank by reliability, and the largest ones are a couple of parents that are probably very well tested, and a few kids that obviously have phenotypic responses. And I can sort my solutions, or in this case, the breeding values, by largest to smallest, and I can identify which one are the individuals that have the largest breeding values to be selected as parents, forward selection, and the kids, and I might have some parents coming along. I don't see there. Here is a parent that I might want to select, and that will be my backward selection and so on. So this is what it will help you to make that decision with the breeding values. Now, I also have the dominance effects, which are the SCAs. And remember, it's not totally the dominance. I will need to multiply by 2 to get the actual dominance effect associated with the family, but SCA is good enough. And you have something very similar where you have values, and this is the deviation of the family with respect to the mean of the family, okay? So they are deviations with respect to the mean. And something similar you have with the epistasis, which are going to be the left deviation. Now, what I'm going to do on the next few things is I'm going to say, well, I already selected the best parents. I can select the best parent. Can I select the best families? And the answer is yes. You can select the best families. And what you need to do is add the breeding value of the father divided by 2, breeding value of the mother divided by 2, sum up, and then you get the answer, okay? Now, in order to do that, I need to do a little bit of manipulations. And I'm only going to do it with 16 families, okay? in this case, what I'm doing is just trust me on what happens here. You can look at the data. I'll provide the data set to explore a bit later. Is actually, I'm going to show you. I'm going to calculate the family effect, or a BLAP effect if you want, or whatever, of the cross. I call it from the cross, which actually is half of the breeding value of the mother, half of the breeding value of the father, which is the GCA of the female, GCA of the father, plus the interaction of that specific family. And I only have effect for 103, 104 families, which is what I tested here. And then you can look at that and you can rank the families. So if I was really interested on selecting this family, I know this family is good, the parents are good, they don't have too much of a family deviation SCA, but you can see, for example, there is a group of families, I remember identifying one, depends on the magnitude, but 0.36, actually this 0.70 is a good deviation of that specific family, a specific combining ability. And this allows me to select the best family, maybe repeat them, and then from there I know I'm going to have to select the best clones or so on, depending on your breeding strategy, okay? Now I can also select the best clones, and then the code, this is very similar. I'll just show you the last bit, where I need to add 1/2 of the breeding value. So this is the total genetic value based on this full model. I need to add the father breeding value divided by 2, the mother breeding value divided by 2, the family SCA, and the specific effect, which is the kind of epistatic effect of that individual. And that gives me the total genetic value. And from there, I can rank the best clones. Now, this is not very different than my previous analysis. I find that the clone 001, 003, so there's a few things here. Well, there's lots of more names on this, are specific good clones. And you can see that this good clone is very good because it has a high non-additive effect. But there is a few clones here that don't have very high non-additive effect, and it happens to be that they are from a very good father and a relatively good mother, and they have a positive interaction of the family. So they are already coming from, let's say, a good background and so on. So this allows you to select. Now, I'm just going to compare this with my original clonal analysis. And you can see the plot between my original clonal analysis and the new analysis, and you see a little bit of differences. The correlation is actually 0.955. These differences are because some individuals have effects that deviate them. So what happens is when you fit this non-additive model, you're estimating every individual component, and some of the individual components, particularly the additive effect, it's better estimated because now you're using all the potential relationships that you have that individual has. And also the dominance is associated with all the clones that belong to the same family. So there's minor adjustments on the clonal value based on this. Now, assuming you are able to split everything together. Okay, so there are some differences on here. Okay, so I'm going to move to, we don't have much time, so I'm going to move to a little bit of the genomics, and then we'll come back to the part of the a few more questions, okay? Now, in the case of the genomics, and I'm going to go very brief with this because I'm not presenting an example with all the genomics, but I just want to give you the flavor. You are aware that GBLAB in comparison to PBLAB, it replaces the additive matrix by a genomic matrix, and we use the same logic here. So I can replace, and I can use the GA, and I can use a dominance matrix also. Now, both of them will be estimated based on molecular markers, and there's many papers and libraries that allow you to do that. And you replace some of the components, so I'll show you 2 variants. Now, some of the epistatic components are calculated. Notice here you have AA, AD, and DD because you have interactions with additive with dominance, dominance with dominance. And in order to do that, you take your original GA and GD, and then you do these Hadamard products, okay? So you can just decide to fit only one component for epistatic, and you see that in some papers, or all 3, and then you have to build the G matrices and their corresponding inverses for each of them. Now that creates a few issues, which I'll mention later, but the main thing is there is a lot of competition between these matrices because they are very similar. especially if you don't have too much of a structure on your population on relationships, okay? And so often it gets very complicated to estimate all the components. There's a lot of confounding and so on. Again, there is a few manuscripts. I've been involved in a few of those a few years ago. And this is something that it has been kind of like quite widely used these days because we can try to estimate non-additive effects from some breeding programs based on molecular data. The GA matrix, this is just a Young formulation. You have the VT-seca and the Su formulation for the dominance matrix. They all have their goods, pros and cons, etc. And there's a few libraries that can do the calculation. A GA matrix is a very good one. And ASReMl is the one that we develop and we use to kind of prepare the matrices for to go into ASReml-R to do some QC and so on. So I'm going to show you the two options. One option is just as crude as saying, well, I had my A matrix before. I'm just going to replace it by the GA matrix. Okay, and that's what I'm going to show you. Just replace it and then everything is the same. But notice that F is still there, is the family. And then the clone within family is going to be the same. Okay, and I'll just show you quickly. This is what I have here. Now the G matrix has been calculated by the author, so I don't have access to the raw data, but I do have access to the GA matrix, which is here from almost all the same individuals. There are 4 individuals that in the phenotypic data that are not genotype, which means I need to subset the data. And this is what I'm doing here. I'm subsetting the phenotypic data to drop those 4 individuals that they're not defined in this genomic matrix. And then I'm going to do a little bit of a blending of the GA matrix. So it's more stable and inverts a little bit better. So I'm just averaging by identity on 2%. And then I get the inverse as required for ASReml, and it seems to be very well conditioned and so on. So it's a decent matrix. Again, I don't know the details of how this was calculated in the original paper. Now, these are my variance components from the full model that I used before. And what I'm going to do now, instead of using this A inverse, I'm replacing it by G inverse, and the models is exactly the same. I'm just calling it model clonal full G. It fits relatively quickly. The variance components, and I'm going to show you, change a little bit. Of course, now this is 3.55 and it was 3.50. That's fine. The standard error actually reduced, which is common to be on genomic data. And notice that I still have 4 clones that are being dropped, so the comparison is not totally true, but everything else seems very similar. I can go back and estimate my dominance, broad sense heritability, and epistasis, and all of them are a bit different than before. The standard errors are lower, but everything seems to be very, very similar. Okay. So somebody is asking, should we still include the ID genotype term even with epistatic effects to avoid pseudo-replication from clones? Actually, I have true replication of the clones. It's not pseudo-replication. So I have each of the clones replicated 3 times. So I don't have that problem with the pseudo-replication. What it is true is that the epistasis cannot be separated completely from the error, and there's always some levels of confounding. Okay, so notice this is not an augmented design with a single copy per clone or an unreplicated trial. This is fully replicated. It's 574 clones replicated 3 times with a pedigree that is quite deep. Okay, so that's what it is. Now, the other approach, and I'm just going to jump back to the slides, and I'm almost finishing with this, is option 2, which really means you estimate the additive effect by individual, dominant effect by individual, and the different epistatic effects by individual. Okay, and that means on A, I associated with a GA matrix, on D, associated with a GD matrix, on the epistatic AA, associated with a new GAA matrix, if you want to call it GAA, and then the same or AAD and so on. So you have all these effects to estimate. And when you fit this model, again, you're going to have 1, 2, 3, 4, 5 genomic matrices, which is quite demanding on data, but it's still possible. Again, maybe not everything is going to be very well separated, very orthogonal, and so on. You have to explore. You might have to drop some terms. There's a chance that you might run a bit of out of memory and so on. Now, what you have on the A is the variance of A. D will be viewed directly, the variance of D. and the sum of these three elements will give you an approximation of the epistatic. Now, notice that the epistatic goes with 3-order interactions, 4-order interaction. So it's an infinite number of terms that you can put. But in this case, we are ignoring the rest and throwing them into the error, which is not totally correct. And again, that's what sometimes implies that you have a few difficulties on this interpretation. Now, you have breeding values for each individual, dominant values for each individual, and epistatic values for each individual. Now, it's interesting that these are not done by family, they're done by individual. So the dominance effect of a given individual is how that individual on average is going to interact with the rest of the individual to express to its offspring a boost up or down. It's not associated with a specific family. Okay, this is similar to the breeding value. Remember, the breeding value is how your additive has behaved when you are being crossed with the rest of the population. Okay, so this model, I can't fit it because I don't have the data, but I'm just showing you here how you will do it. If you have the G matrix, you have the D matrix or GD matrix, and you put the inverse associated with effects for additive, effects for D, for AA, for AD, and DD, and then obviously you have to pull all those effects and sum them up to get the total clonal value, or you focus on the ones that you're interested on. Okay, so that's the way to fit. I mean, I can't fit it because I don't have all these matrices. I don't have access to the raw data, but with those libraries, it's relatively easy to do. Okay, so just a few closing remarks. I'm almost running out of time, but there will be time for a few questions. I do like this model because it gives me the genetic architecture of a given trait. It tells me what's going on underneath. That's not necessarily easy to fit. And in some breeding programs, you have to create this sort of experiment with the data, with a good crossing and with everything. So it allows you to do this partition. I find it extremely important for some breeding programs. I mean, when I work in eucalyptus or things like that, it's very important, or in sugarcane also, to know is the main genetic variability on the additive and the dominance on the epistasis. Do I need to worry about the epistasis, etc.? And I mentioned a few of those cases. It also allows me to estimate the total genetic variance, which is normally what I care, to select the best clone. So if it's there, it's fine. Now, this is not a model that is easy to fit. Again, I'll talk a little bit more about it later, but because you need a good structure. Now, the information it gives me is tremendous amount of information because I can select the best parents, mother, father. I can also select the best families, and I can actually do forward and backward selection to select the best kids from their additive values, select the best crosses, and select the best clones at the same time. So it adds a lot of value. So that's what I'm mentioning about this. Now, it has difficulties, and I mentioned some of them about the male cross with several females and so on, but I also mentioned that you need several clones within a family, and they need to be replicated, okay? And again, sometimes you still have issues with the components and so on. Those issues tend to disappear a bit more once you use your genomics data. Okay, but I think it's a tremendous model to use. And if you have genomic data, you can use both option 1 or option 2 that I presented before, and then see what happens. But this requires a bit of a trial and error. Okay, so I'm going to finish there. And I'm going to see if there's a few questions. And if you have a few more questions, just pop them up. Again, you can always find us through different ways, my email or through support to have a few more questions, okay? So let's see, a couple of questions and then we'll be finished.
So there are a few more questions that we didn't have time to go around to. One of them was, what are your thoughts in using additive model in a natural population with population structure?
That's a very good question. It's going to be hard. And actually, it's going to be easier if you have genomics data, because normally in a natural population, you won't have the crossing that you expect. So parents might be only crossed with one female. And that means you need to look at the relationship of that parent with the other parents. Now, you can improve the quality of this. I know, for example, in the case of sometimes in the case of fish or in the case of birds, you have several males, or you might have several hatches from a given bird over time, which will mean different males, and that allows you to kind of combine the data in a population. So it's harder, but it's not impossible. And again, as I mentioned, you might not be able to split dominance from epistasis and have to clamp it up together. It's just a trial of error, but again, it's still risky. Okay.
And I think we have time for another question. How would you estimate non-additive effect using H matrix in genomic selection? Would you just use IDV entry ID?
So depends on which approach you want to do. So you can replace the A inverse that I had on the original model. This is this full model. Let me just go back to that full model. In this full model, you can replace this by an H inverse that you have. And obviously that's calculated with the pedigree and genomics and so on. Now, if you want to go to the full model where you have additive dominance and epistatic matrices, you have to calculate those additive dominance and epistatic matrices, and the library AGH matrix allows to calculate those for pedigree and for genomic, and then you have to combine it obviously as an H matrix. Okay. There's one question I don't have very clear is somebody's asking, this will work with 4X. I don't know what that 4X means. Sorry. And maybe tetraploids. Oh yeah, of course, this will work for tetraploids. I was 4X and I thought it was the depth of the genotyping. It can work with tetraploids. I mean, actually, strawberry is an octaploid, but we're assuming it's a diploid. It is how you construct the matrices, the genomic matrices or the pedigree-based matrices. Now, you have to be careful that when you are with higher order polyploids, you're more likely, for example, the dominance matrix from polyploids is very likely to be very similar to an identity matrix, because the probability that two individuals are exactly the same pair of alleles from your, let's say, octaploids, it is very small, so that those higher order matrices or the dominance matrix tend to look a lot like identities. And when they're identities, you can estimate effects. Okay, well, I'm going to stop here. I know you have other questions and I haven't answered all the questions. I'll make sure I go through them. And anything else you want to add, Valerie?
No, thank you very much, Salvador. That was very interesting. And thank you everyone for joining us today. So you should receive the follow-up email in a day or two with the link of the recording, the slides, the code, and the unanswered question. And please feel free to share the recording with your friends and colleagues if you wish. And I think that's it for now. So enjoy the rest of the day, everyone. And thanks again for everybody, and to you, Salvador.
Thank you very much.